Great. Thank you very much. First, let me say thank you to Mr. Miyaoka-san and Sanyo-Trading for inviting me to speak today at this webinar, and for everyone's attention who is attending today. I'm looking forward to sharing Spectradyne's technology with you, as well as some of the areas in which our customers use it to accelerate their research. My goals for this webinar are twofold. First is to encourage you to think about the importance of measuring size and concentration of the particles in your samples. This is a serious question, and I hope to convince you of that at the beginning of the talk. And then we'll spend also time asking you to think about whether your measurement is doing a good job, whether the measurement method that you're using is working properly and operating within the bounds that it's supposed to be. So in the first part of the talk, we will review why particle size and concentration are important metrics, review a few of the most common measurement techniques similar to Mr. Hiroyuki-san did earlier. I will give some detail about how Spectradyne's technology specifically works to accomplish the particle analysis that it does. And I will share with you a few examples where that highlight the value of perspective. So comparing different technologies for measuring the same sample to understand the differences and the pros and cons, the benefits and the drawbacks of each technology. After this, we'll all be on the same page from a technology perspective, and I will share a few application highlights where Spectradyne's customers are leveraging the power of our technology to enhance their science and their research and their products ultimately. We'll cover 4 areas. The first is exosomes and extracellular vesicles. Second will be nanomedicine, specifically lipid nanoparticle samples. Then a quick example from virus quantification, how the Spectradyne's technology enables very fast virus quantification. And then I'll share an example from biologics formulation development also, where Spectradyne's technology offers significant time savings in stability assessment of different formulations. So why is size so important? Well, ultimately, the size of the particles impact the safety and efficacy of a drug product pretty directly. So let's talk about how. The particle size determines the diffusion rates of the particles and the clearance rates in a biological system. So how quickly a body clears out a therapeutic, for example. This is essentially the bioavailability of the drug, how much of the drug's available for uptake. And this is directly related, obviously, directly impacts the drug efficacy. Size also determines the volume of each particle. And that in turn really defines the volume of active ingredient of payload that can be carried in each particle. So it's kind of like a single particle dose. Knowing the size tells you that. Ultimately, the size of the particles in the sample are critical for the safety and the efficacy of the product. What about concentration? Well, many therapeutics now are based on nanoparticles. For example, the 2 most prominent vaccines here, anyway, in the United States, are from Pfizer and Moderna. These are LNP lipid nanoparticle-based vaccines. And in this case, the concentration is directly the dose of the drug, right? So it's imperative, very important, to measure the concentration in order to deliver a safer, more effective product. Must be carefully controlled. So let me walk you through a scientific example from a researcher's perspective to illustrate this. Suppose you have developed 2 different therapeutics, A and B, and you want to compare their efficacy, right? Which one is better? So we start by preparing the 2 different formulations. We apply them to a test system. Maybe it's an animal model, or maybe it's in vitro cell culture. And then we compare quantitatively the results from the 2 different formulations. And as a scientist, you would never think to do this experiment without first controlling that the amount of product A and the amount of product B that were administered were the same. It's critical, right? So the dose and so the accurate size and concentration are critical for performing good science. Let's review a few of the quantification methods that are currently commonly being used. The first is dynamic light scattering, and this builds on what Mr. Miyaoka-san was describing earlier. Dynamic light scattering is an optical technique, as he mentioned, and importantly, it's ensemble averaging. So it measures many particles at once and can only return an average from that. In addition, as he mentioned, dynamic light scattering does not give any information about concentration of the particles. So we're not getting dose from this technology, but it's very commonly used, so we'll talk about it in more detail in a minute. Another technique, as Mr. Miyaoka mentioned, was nanoparticle tracking analysis. This is another optical technique. In fact, it's very similar to dynamic light scattering, which we'll discuss. And more researchers are discovering its limitations recently as it's been in use more. And one of them is critical, which is that the limit of detection, the smallest size of particle that can be detected, really depends on the sample. And that's a problem if you're measuring samples that you don't know already what's in there. So we'll talk about that in more detail. I'll share some examples. In terms of non-optical techniques, there is one called tunable resistive pulse sensing, or TRPS. This is a non-optical method. It's electrical, so at Spectradyne, we're a fan of that approach. However, it's difficult to use in its implementation. It's a little bit simplistic. And that brings us to Spectradyne's technology, which is called microfluidic resistive pulse sensing. This is a powerful technology that's very fast and easy to use and affordable. And let me give you a minute describing our product and this product and exactly how it works, so because it's new and maybe you're not familiar already. So this is a picture of our instrument. It's called the NCS1. It's smaller than it looks in the picture. It's only about 30 centimeters wide on a side. This is a brand new technology that delivers accurate concentration and size starting from about 50 nanometers in diameter up to as big as 10 microns. So it really spans the gap between liquid chromatography technologies at the very small end and flow imaging technologies on the very large end. As I mentioned, it uses an electrical detection method, so it's orthogonal. It's a fundamentally different technology than most of the optical techniques, and it's fast and easy to use. Typically, results are obtained in a few minutes. And because it uses microfluidics, this is a microfluidic cartridge that we use for running the samples in the instrument. That delivers many features, including that only 3 microliters of the sample are required for analysis. Many of our customers find that to be extremely valuable when they're working with precious samples. Maybe they're hard to make or they don't have a lot of them available. One of our customers measures the saliva from insects, ticks. They're very small insects, and you can imagine that can only be done on a technology that requires a few microliters of sample. Okay, so let me explain how this works. So microfluidic resistive pulse sensing is really just a modern version of the Coulter principle, which has been a gold standard for measuring large particles and cells for decades. If you've had your blood analyzed for cell counts at the hospital, you've probably already benefited from this technology. In this technology, particles are measured one by one very quickly. So the way this technology works is we pass the sample through this constriction in the fluid flow. So we flow the sample through a bottleneck in the constriction. And every time one particle goes through this constriction, we get an electrical signal that looks like this. So this is actual data showing the measurement of a single particle as it passes through the orifice here, the constriction. And every time this happens, we measure 2 things. Okay, one is how big the particle is. That's given by the height of this peak. And the other one is how quickly the particle went through the constriction. That tells us how much sample we're measuring. So this is the signal from one particle. If we measure for longer, this is showing a longer data acquisition. You see many, many peaks over time. And you can see that the size of the peaks are grouped in 4 different size groups here. We can see that directly from the histogram of counts of particle sizes. But we can do better than that because we know the volume of the sample that was measured. So that means we can convert these counts to absolute concentration, so the number of particles in a given size range per volume of sample. And so this is what the results of that particular sample measurement are. This sample was a mixture of polystyrene beads, 50, 90, 120, and 150 nanometers in diameter. And you can see that our technology clearly resolves all 4 components of the sample and can be quantitative over any size range. So we can measure the relative or the absolute concentrations of any one of these populations in the sample. This combination of accurate concentration and size with ease of use is currently not available on the market from any other method. Let me share with you 2 examples that show the performance characteristics of the technology. First is the linear response in concentration to show the accuracy of the concentration measurements. So these are 4 different measurements of a sample that was a retroviral drug product. So these are virus particles in the peak here that are all well-defined size here. And we did a serial dilution of this sample. And when we measure the concentration, we get a very nice linear response, as you would expect and demand from your technology. The second example I'd like to show you illustrates the broad measurement range of the technology. So this is a measurement of a liposome or LNP lipid nanoparticle sample. And you can see we're measuring the particle concentration on the y-axis versus particle diameter. And the diameter is spanning from 60 nanometers up to 6,000 nanometers, 6 microns. And over that size range, the concentration of particles in the sample spans over 9 orders of magnitude. This is a huge dynamic range, and this is one of the only technologies that can do this. So let's shift gears now to the second question for my second goal for the webinar, which is my measurement method doing a good job, and am I operating it in the appropriate regime for this technology? Another way to ask that is, how well do I know the limitations of this technology? And let's start by talking about dynamic light scattering, because in the nanomedicine world, for lipid nanoparticles, this is still a very common technique. Even though it can't deliver any concentration measurement, many people are familiar with this technology in this space. So let's get on the same page about how it works. And Mr. Miyaoka-san explained this already a little bit. I'll repeat a little bit quicker what he said. So we start by illuminating the sample with a laser. And remember, we're illuminating many, many particles at once. So this is a bulk measurement. And we're detecting the light that gets scattered from those particles to a detector. Now, the particles are not standing still. They're doing Brownian motion, right? And by monitoring the dynamics of this scattered light, so the changes in that scattered light over time, we can infer the Brownian motion characteristics of the particles in the sample. And we can infer then further, we can infer an average size of the particles. And this is called the Z average of the sample measurement. However, there's a limitation here, and that is that small particles scatter much, much, much less strongly than large particles. In fact, the dependence is that the scattering intensity is proportional to the diameter of the particle to the 6th power. So for example, if we say that a particle with diameter 50 nanometers scatters 1 unit of light, then 2 times bigger particle scatters 64 times more light, and a 4 times larger particle scatters 4,000 times more light. What this means is that in a real-world sample, when there are a range of particle sizes in the sample, the dynamic light scattering measurements are heavily biased by large particles in the sample. Let me show you an example like that. So this is a real measurement of DLS of a real sample, okay? And you can see that there is a peak in the scattering intensity distribution, and it reports a Z average value of 165 nanometers. So, you know, if you're a scientist and you've just made a formulation that is supposed to be nanoparticles, then you're pretty excited about this because it looks like there's a well-defined population of particles about 165 nanometers diameter. However, you are a careful scientist and you look back at the international standard for using this technique, and it says a few very important things. First is, this is an intensity distribution. It's not a particle size distribution, right? So it does not mean that the distribution of particle sizes in the sample looks like this. All we know is that they scatter as an average like 165 nanometer particles. Second, right here in the standard, it says that converting this scattering intensity distribution to a particle size distribution is a bad idea, basically, okay? It really says fraught with danger and ill-advised. So it's trying to scare people from doing it, right? So what do we really know about this sample? Well, it turns out not that much, right? We just know the Z average of the measurement. The size distribution that I showed you before is not meaningful. Why is that? Well, the reason is because many different size populations, the size distributions can lead to the same Z average measurement. For example, we would love for the sample to look like this, right? It's a well-defined particle size distribution with a center at 165 nanometers, right? But remember, we're not getting concentration information from the dynamic light scattering. And so it could just as well come from a distribution that looks like this. Still, that would not be bad. It's got a well-defined peak size, right? But it could also come from a more complicated size distribution that averages out to 165 nanometers, remember, with a bias towards picking up the larger particles. So dynamic light scattering really is telling us neither the particle size nor the concentration in the sample. And we need an orthogonal measurement technique, a fundamentally different technique, in order to know what's really going on in the sample. And so, of course, we did just that. When we take this sample and we measure it on the NCS1, this is the true size distribution of particles in the sample. And we know that because we made the sample. It's a mixture of polystyrene beads. This is a very, very simple sample of 50, 94, 122, and 150 nanometer particles, all mixed together at approximately equal concentration. Spectradyne's NCS1 delivers the high resolution sizing and the absolute concentration on any size range in here that you need in order to actually find out what's in this sample. So to summarize, dynamic light scattering always gives an average particle size, which is great for monodispersed samples, and it's a very useful technique for this kind of material. However, when you get to polydisperse or unknown samples where you don't know in advance what is in there, it's not really telling you that much about the sample. And it's really important then to use orthogonal techniques to measure the actual size distribution and of course the concentration, because the concentration is the dose. Okay, let's keep going and talk about nanoparticle tracking analysis for a little. It's still used by the majority of extracellular vesicle researchers. So let's review how this technology works. It's very similar to dynamic light scattering. It uses the same fundamental principles and ultimately suffers the same limitations, but we'll talk about that. So, excuse me, we start by illuminating the sample again with a laser. As before, the particles are not standing still, they're undergoing Brownian motion. And Mr. Miyaoka-san had a very nice illustration of this same idea. So particles are doing random walks around in the solution, and small particles make larger excursions, and large particles stay more still. They undergo less visible Brownian motion, right? So instead of now measuring the dynamics of all the scattered light in bulk, what we'll do is we'll take a video of individual particles. the scattered light from the individual particles, and we'll track these random walks around in space under a microscope, essentially. And from that, we'll infer particle size from individual particles, right? So that's a great idea. And in principle, now we're measuring single particles in this technique, and that's great. However, there is a limitation, and that's the same as for DLS, dynamic light scattering. And that is that the smallest particles scatter light so much more weakly than the big particles that they get obscured. We can't see them in the video, basically, okay? So when NTA is used to characterize, again, broad size distributions, the large particles are too bright for the camera, and they hide the small ones, and we can't see them in the sample. Let me give you an illustration of that. This is a very easy experiment that if you have an NTA instrument, I encourage you to try because it's very instructive. What we'll do is take an NTA and we'll take a few different samples. First is a monodisperse population of polystyrene again. These are 208 nanometers diameter, okay? NTA does a great job of measuring the sample. It shows a nice clear peak, and the concentration, here I've normalized it out, but it's about right, okay, for these particles. We can do the exact same thing for a 150 nanometer particle, monodispersed sample measured all by itself, and NTA does a good job again. We can do the same thing for 94 nanometer particles. And from this, you would think that NTA is doing a great job and that the sensitivity limit, the detection limit, must be much smaller than 94 nanometers diameter, right? Because it picked these up just fine. In fact, you would be right if the sample is monodisperse. NTA has been shown to measure as small as 10 nanometers with very brightly scattering gold particles, for example. The key word is that these particles are monodispersed, so they're all the same size. So here's where we put a twist in the experiment. Let's mix these three samples together and make a polydispersed sample, right, with a range of different sizes. When we do that, this is the curve that we measure. So what's happening here is that we don't see the 94 nanometer particles. They have totally disappeared in the measurement, even though they're in there at an equal concentration as the other ones. What's happening is that the big particles in the sample are dominating the images in the video, and the small particles are too dim to be detected. What we've shown here, and this is the scary part, is that sensitivity, the sensitivity limit of NTA technique depends on the sample composition, right? And that's a major problem if you don't know what is in the sample to begin with, right? It works fine for monodisperse samples like this, but as soon as you're measuring a sample that you don't know or has a broad size range of particles in it, it's not going to give you the answer that you need. And so, and I'll show you another example, a real world example of this a little bit later. What happens when we use an orthogonal technique to measure these samples, like MRPS? We repeat the same individual experiments and they all measure just fine, the same concentration. This time, when we mix them together, we get exactly what we expect. We get a superposition of the three monomodal distributions, equal concentrations, and resolved the different components. Okay, so to summarize NTA, NTA is using Brownian motion again and light scattering to measure individual particles, right? That's good. And for monodispersed samples, it works just fine. And you can get actual concentration information from that. Problem comes when we're looking at polydispersed or unknown samples. And in this case, you really don't know how to interpret the data that you're getting from it because you don't know in advance what's in the sample. And again, we have the same conclusion, which is we need to use orthogonal techniques to measure the actual size and the concentration of the samples. All right, so I hope that was clear. And now let's move now to the application highlights section of the presentation. So here I'd like to just give an overview of 4 different areas where Spectradyne's customers are finding real value for our technology, helping them improve their science and do faster research for better products. So the first area I'd like to highlight is in extracellular vesicles, or EVs. And here, really, it's about controlling the experimental variables. It's pretty straightforward. Measuring the accurate size and concentration of the particles leads to better science because you can control the mouse experiment or the different biology experiments that you're operating in better and reduce experimental variability. Now, you don't have to take my word for this or pretty much anything in this talk directly. We have many published articles available on our website. And one I would like to draw your attention to on the topic of extracellular vesicles is this one from Johns Hopkins University in Baltimore. It does a very nice job of comparing 4 different single particle analysis techniques for characterizing extracellular vesicles. And it does so in a very even-handed, we feel, unbiased approach. It's a nice article. So by very well-respected researchers there at Johns Hopkins. So I'd encourage you to go check this out, read this paper, or contact us to learn more about this. So let's start again with NTA because it's very popular in the extracellular vesicle world. And we'll take it again from a scientist's perspective, where I've measured this EV sample, purified my EVs, and I've measured them by nanoparticle tracking analysis. And this is the data that I received. Okay. If you're a scientist and you see this data, you get pretty excited because we've got a nice peak in the size distribution. It looks very promising. And this is an easy number to track, and you can get a number from it, and we can measure the concentration. This is a very easy result. Let's look at it in a little more detail because we're careful scientists, right? So the first thing I'll do is plot the exact same data just on a log Y scale. Okay, so we can see the full range of concentration here. So this is the same exact data, same peak that we just looked at. As a careful scientist, you take the same sample and you measure it on an orthogonal measurement technique. Luckily, you have one of Spectradyne's tools in your lab, and you measure it by MRPS. And here you see something very different, right? Here, now the particle size distribution keeps on increasing with a roughly power law size distribution, dependence of concentration on size, all the way down to the detection limit of this technique, right? And so what's happening here and which one is right? Well, let's confirm by measuring it a third time with the gold standard for EV imaging, which is transmission electron microscopy, or TEM. And what we see here is that MRPS agrees almost perfectly with the TEM, and certainly the distributions agree, exactly, right? What's happening here is exactly what we talked about before. We have a broad size distribution of particles in the sample, and NTA is losing sight of the smallest particles in the sample. So the counting efficiency, or the sensitivity, is rolling off at smaller sizes. Unfortunately, in this sample, what that means is that NTA is off by 10,000-fold in concentration in this size range here. Remember, this is a log scale, so this is 10,000-fold difference between the true distribution and what NTA is measuring. And this is the critical size range for extracellular vesicles, between 50 and a few hundred nanometers. NTA is missing, 10,000-fold in concentration. This is a major eye-opening experiment. The conclusion is that orthogonal measurements are critical for accurate particle analysis, right? And this is true across the board in science generally. Okay, so let's take another example now. This is a nanomedicine application, specifically lipid nanoparticles. And here, Spectradyne's customers use the NCS1 because of what we talked about at the beginning, because it delivers more accurate dosing and better controls for the science, for the scientific comparisons and experiments that are going on. Concentration is a critical experimental variable that needs to be controlled accurately. So here, we'll look at 3 different nanoparticle formulations, lipid nanoparticle formulations. They're slightly different recipes. And the first thing we do is measure them by dynamic light scattering. So sample 1 returned a Z average of about 66 nanometers diameter. with polydispersity index 0.1. Sample 2 measured about the same Z average, right? These are pretty insignificant differences with a similar polydispersity index. And sample 3 could not be measured with dynamic light scattering, presumably because the polydispersity index was too high. So what's going on in these samples? Well, we took the samples and measured them on the NCS1. And this is what we see. So now we're showing samples 1, 2, and 3. And remember, these are now actual particle size distributions because we're measuring particles by counting them and measuring their size one by one. So we've got concentration on the y-axis and particle diameter on the x-axis starting at 50 nanometers or so. First, we see that there are clear differences between the three populations, right? Sample 1 is in the middle. Sample 2 shows a smaller mode to the size distribution. And sample 3 has many small particles, but a much larger prominent peak in the size distribution. These are very rich distributions too. I'll point out that the blue sample 1 shows a hump out here with a higher concentration of large particles when compared to the green or the red samples, for example. And these are meaningful differences for when you're talking about the bioavailability of these lipid nanoparticles, how easily they will distribute in the body, and ultimately the efficacy of this product. As a reminder, I'll show you what dynamic light scattering measured for us. In the blue and the dashed lines, we're showing the DLS Z average. And remember, it was not really able to distinguish samples 1 and 2 at all. Not to mention, it couldn't measure sample number 3. So this is an example in lipid nanoparticle space and nanomedicine where MRPS and counting these particles one by one is really giving a much richer picture of what's going on in the sample. And this can be used to feed back into all the development process and ultimately do faster R&D, right, and lower cost. So to remind you, just to summarize these results, we're measuring 3 different peak diameters, which are significantly different. We can be quantitative in concentration over any size range here. These are the number of particles that were counted to return the MRPS data. So over 10,000 particles measured in each of these examples. And dynamic light scattering is basically unable to distinguish between these samples at all. All right, so let's move to the third application I would like to highlight, which is an application in virus measurement, and which is, of course, relevant to gene therapy applications. And here, our customers are using MRPS to quantify the viral titer in a few minutes, as compared to a few days sometimes to measure the virus titer by cell culture methods. So this is a huge time-saving opportunity for those working with viruses, and let me show you an example. This example is human adenovirus 5. And I wanted to share with you what the raw data looks like in this sample. So remember, I was explaining how the technology works, and I said there was a spike, a peak, every time a particle gets measured through the constriction. And so what we're seeing here is about a second of acquisition time from the instrument. And all of these particles up here are the virus particles themselves. So we're counting them one by one, and you can just see them being measured this way. When we analyze this data and turn it into concentration measurements, this is what we get, which is a concentration versus size plot again. And here I'm showing you 3 technical replicates of the measurement. There's a very clear peak right where it's expected, around 90 nanometers for this adenovirus 5. And you can see excellent agreement between the replicate measurements. Not only that, it's quantitative, right? We can measure the concentration over any size range here. So you can see that when we integrate just over the peak of the virus peak in this sample, we get very good agreement on the particle concentration, on the virus concentration. And as a reminder, these are absolute concentration numbers, so it's particles per mil. It took about 5 minutes per sample to make the measurement, and no tissue culture was required to do this analysis, right? Compare that to if you've done viral titer in a biological system before, you know that you need to first purify your sample. You need to plate out your cells. You need to serially dilute your sample. You need to incubate the virus with the cells, and this can take days, right? And then you need to count plaques, and hopefully you get one that's in the right countable number of plaques to give you the accuracy you need, and you need replicates, right? This can literally take days, and especially in human cells. And here we've got the same data in just a few minutes per sample. If you don't like adenovirus 5, we have many, many other examples of virus measurements we've done on NCS1 and our customers are doing. And if you don't see your virus on here, we'd love to hear from you and measure your virus too. So let's work together. Please contact Sanyo-Trading to arrange a demonstration. Okay. Finally, I would like to share with you an application highlight in biologics formulation development. So here, the situation is that a biologic drug has been prepared, formulated, and it's being tested for stability. And typically this happens by taking the sample and stressing the sample with heat or some other form of stress for a certain period of time, and then characterizing the number of particle aggregates, usually protein aggregates, in the sample over time to understand how stable the drug formulation is. This is a very long process, and many times the stress period can be for days or weeks on a shelf or something like that. And so people working in this area really want faster methods to be able to assess the instabilities of a formulation. What I'm showing you here now is measurements on the NCS1 of the same formulation stressed for different time periods. So there's a 0-minute stress, so this is the control sample. There's 10-minute stress, 20 minutes, 30 minutes, and 60-minute stress. And it's very clear, so here we're looking at concentration again as a function of particle diameter, right? It's very clear that there's a trend. More stress leads to more particles, right? More aggregates in the sample, and this is expected. But note the short times here, right? This is a maximum 1 hour stress time, and already there's a very big signal here. We can do better than that, though. So let's look at what happens to the concentration over different size ranges here, and this will help make a more general point that I'm trying to make. If we look at the large particles in size range number 3, these are the large particles, we can plot the concentration as a function of stress time here. So there's the 0 time, there's 10 minutes, 30 minutes, and 60 minutes, right? So we see, a slight increase in the concentration of particles in range 3 over this time period. If we look at size range 2, which are slightly smaller particles, we see the same trend, but a slightly larger signal, right? There's a bigger change in the concentration as we go to smaller sizes. And that holds when we go to the smallest sizes, where when we look in the range of 150 to about 200 nanometers, you see a very large signal change, even at very short times. And this is the point that our customers understand very well, which is that by looking at smaller particle sizes, the point here is that at smaller sizes, we can detect aggregation sooner and with less stress time, right? We get a bigger signal by looking at smaller sizes. So this is what our customers understand very well about our technology, which is that the smaller you can detect the aggregates, the sooner you can detect the aggregation, right? And detect an instability. So they save time and ultimately money by detecting particles when they are smaller in size before they wait for them to aggregate to much larger sizes. Hopefully that makes sense. I'm sorry for the interruption. Okay, so at this point, I will conclude, I'll summarize what I said and leave you with a few thoughts. So one is, I hope I've convinced you that that you should take size and concentration measurements seriously, right? It's the concentration is the dose, and the size is the efficacy and also the dose, right? Very important. Secondly, I hope that by sharing some examples of comparing the different technologies, that you've been able to ask yourself the question, is your measurement method doing a good job? And are you operating within the appropriate bounds of those technologies' performance range? So I hope that you will take away from this that accurate size and concentration are important because they're critical parameters for good science. To understand your measurement techniques and ultimately look at your samples with different kinds of technologies using orthogonal techniques in order to get a complete picture of what's in your sample. We are available online as much as you like. Come visit our website at Spectradyne. It's nanoparticleanalyzer.com. We have information about all the applications that I described today, plus others on the website. There's information about our products, specifications, as well as new technologies as we release them. We have a large and growing library of publications and videos and application notes that, that highlight these and other applications, and also come from independent, there are many peer-reviewed journal articles on here, so you can get other people's opinions. And we have a blog that we update as frequently as we can, including analysis of COVID vaccines. So I encourage you to come and check it out. And if you have any questions for us, please feel free to reach out to us directly. So at this point, I will say thank you again to Sanyo-Trading Co for hosting me, and Mr. Miyaoka-san for inviting me to speak today. And I think we will take your questions as best we can. Thank you very much for your attention.